So this video is looking at the second half of the physical factors that affect bacterial growth. And so in this one we'll look at oxygen, water, hydrostatic and osmotic pressure, and radiation. So oxygen is a very important physical factor for ensuring appropriate bacterial growth. And there's a whole variety of needs around oxygen among bacteria. Bacteria that are aerobic, and we sometimes refer to them as just aerobes, they require oxygen to survive and to grow. And it's frequent among aerobic bacteria that availability of oxygen can be a limiting factor for their growth. So if there's not sufficient oxygen, then aerobic bacteria will have a hard time surviving. If it's an obligate aerobe, it's obligated to find oxygen or else it won't live. If it's a microaerophile, these are ones that prefer to have just a little bit of oxygen. So they don't want full-on atmospheric oxygen, but they live somewhere between the zone of no oxygen and full-strength oxygen. We could think of it that way. Okay, so those are their aerobic bacteria. If it's an obligate anaerobe, must have oxygen to grow. If it's a microaerophile, that means that they that's the phile part, little bits of oxygen. Anaerobic bacteria, we can just call them anaerobes, they're the ones that do not require oxygen in order to survive and grow. If it's an obligate anaerobe, then oxygen kills them. But it's usually, or being in the presence of oxygen, I should say, will kill the obligate anaerobe. But it's not really the oxygen that kills them, but it's these superoxide compounds that will form in the presence of oxygen. So an obligate anaerobe cannot stand oxygen. Facultative anaerobes are ones that can survive without oxygen, but if oxygen is available, then they will do aerobic metabolism. So they'll do cellular respiration if oxygen is available, but they can do just fine without oxygen. So facultative, that means that they have the facility to be able to survive without oxygen. Aerotolerant anaerobes are ones that will never use oxygen as part of their metabolic processes. So they don't make ATP using oxygen, but they're not damaged because oxygen's around. So an obligate anaerobe will be killed around oxygen, but aerotolerant anaerobes, they do just fine with oxygen. They just don't need it for any of their metabolic processes. Okay, so bacteria can be around their use of oxygen. We can talk about aerobes and anaerobes. If they are obligate, then they have to have whatever the condition is. If they are facultative, then they can manage being anaerobic, but prefer to be to be able to have oxygen. And so where do we find these guys growing in a colony? So bacterial growth they're showing you is this whitish stuff. So that's the murky area where the bacteria are hanging out. So if it's an obligate aerobe, then they are going to be clustered around the top of the colony or the top of the culture because they're up there where the oxygen is most abundant. And generally when we store or when we incubate bacteria in tubes like this, if there is a screw top lid, then you leave the lid a little bit open. Now, if it's an obligate anaerobe, then we're going to see those bacteria as far from oxygen as possible. Those that are facultative anaerobes, that means that they can handle being without oxygen, but they will use it. They're going to be found anywhere in the culture because up here they'll be using oxygen, down here they won't be. And remember, the microaerophiles are the ones that like it to have a little bit of oxygen. And so they're going to be right here in that zone between the aerobes and the anaerobes. So with moisture, it's a requirement for a vegetative cell. It's got to have moisture. They can only go a couple of hours without having a constant supply of water before they dry up and die. But if a spore has been produced, then that can remain, in the resting stage, the non-vegetative stage, dormant for quite a long while. And we've talked about that, thousands of years even for some. So that's moisture. Hydrostatic pressure, that's the pressure that develops because there's water above. And so the deeper you go in water, then the greater the hydrostatic pressure is, because there's more and more water piled up on top of you. So hydrostatic pressure is the pressure exerted by standing water on the organisms that are at the bottom of that body of water. So for every 10 meters down that you go in water, you add 1 atmosphere of pressure. An atmosphere is something like 14.6 pounds per square inch. That is, if you were at the beach standing there at sea level, that's the air pressure you would feel on your body, 14.6 pounds per square inch. So all you have to do is go 30 feet down and you will double that, and you'll be at 28 or 29.2 pounds per square inch. Go another 10 meters down and you add another 14.6 pounds per square inch. Now the book says every time you go down 10 feet you double the pressure. That is not accurate. They screwed up on that. It's a multiplicative factor. So if you go down 30 feet, it's 14.6 times 30. If you go down 100 feet, it's 14.6 times 100. So that's easy to do. It would be 1,460 pounds per square inch would be the pressure on whatever the organism is there at 100 feet below the surface of the water. So at 7,000 meters, which is about the deepest we have found in the ocean, you see about 700 times atmospheric pressure found at the Earth's surface. So that's a lot of pressure down there. So barophiles, these are bacteria that like that kind of pressure. Baro means pressure. So if you've taken anatomy and physiology, you talked about baroreceptors that are noticing blood pressure in the body, and a barometer measures air pressure. So barophiles like it. They like being at high pressures, like you would find at the bottom of a deep lake or down at the ocean floor underneath, a mile of water. And the reason that barophiles do better at pressure is because they require these high pressures for the enzymes to function. It turns out that among barophiles, if you bring them up, if you bring them up to a reduced pressure zone, so let's say you took something that was at 7,000 meters below the ocean surface and you brought it up to sea level and were experimenting with it, you would find that the bacteria do not grow, or they don't grow very well, because their proteins will denature at low pressure. So once again, it's the proteins and their function as enzymes and as transport mechanisms across cell membranes that when we reduce the hydrostatic pressure on a barophile, then we wind up denaturing the proteins and killing them, or at least slowing their function. Osmotic pressure, on the other hand, is different from hydrostatic pressure. So hydrostatic pressure is that force by all of the water above these organisms. Osmotic pressure, on the other hand, is a force that draws water from the side of the membrane that has the... there we are. I'm sorry, not from. But it draws water toward the side of the membrane that has the greatest solute concentration. So, for instance, if we're talking about out here, so if we're inside the cell, and I'm just making these numbers up, let's say it's a 10% salt solution inside of the bacterium, but you put it into the ocean, which is something like 33 parts per thousand. Oh, I've gone the wrong way. Yeah, the ocean water is about 3% salt. So let's say we have a bacteria that is, the internal environment is about 1% salt. So we put it in a really salty environment. I should have worked this out before I started the lecture, but you're with me now. So when we put bacteria in these salty waters, there's going to be a call for the water to leave the cell, because water is going to go to the place where water is lowest in abundance. So out here it's 96.7% water. In here it's 99% water. So osmosis is going to draw water out. We draw water toward the side of the membrane that has the highest solute concentration. And so bacteria cell membranes, they contain proteins that are capable of regulating the movement of dissolved material from one side of the membrane to the other. And this allows them to survive under a wide range of osmotic pressures. But extremely high concentrations, like high salt or high sugar waters especially, can exceed the tolerance for the bacteria to withstand that osmotic stress. And so what happens is, here's a cell, and here's the bacterial cell wall outside. So this is before we subject it to a really salty environment or to a really sugar-rich environment. Here is that should be the cell wall. Then what we see is the bacteria will plasmolyze, where the cell becomes all kind of crumpled up because we've drawn water from out of it, and we leave the cell wall back where it is, but the membrane now is all crumpled up inside that cell wall. That is a cell that has undergone plasmolysis. So extreme concentrations like high salt or high sugar are going to exceed the tolerance of bacteria for osmotic stress, and they'll plasmolyze. But if you put it in distilled water, on the other hand, so now if we take this cell and put it in distilled water, water will move in, but at some point, the pressure that the cell wall exerts is going to prevent any more water from coming in. And so the cell will just become really stiff as the membrane presses against the cell wall, and it becomes turgid. So what are the characteristics of bacteria in relation to the osmotic pressures that they want to be in? Well, if it's a halophile, halo here is about salt. Halogens, those are the salt. I'm trying to think of halides are salts, H-A-L-I-D-E-S. So that HAL, that means saltiness. So halophiles require moderate, like 3.5%, that's sort of ocean water, to really high quantities of salt, 20 to 30% salt water. And salt, especially sodium chloride, is a common salt that provides this kind of osmotic pressure. And so we find these kinds of moderate to large quantities of salt in ocean waters, and then in captured lakes that have a lot of salt water in them. So the Great Salt Lake, for instance, the last remnant of the great inland sea that used to flood across the central plains of the United States. The Dead Sea over in the Middle East is a place where there's water coming in, it brings salts with it, but there's no outlet, and then the water evaporates and leaves the salt behind, and more salt keeps coming in, and so the Dead Sea is very salty as well. And even we can even find halophiles in commercial brining vats, where sometimes these halophiles will spoil the food products. And so here's an image of the Great Salt Lake, and you can see the salty crust that builds up on the shore there as the water evaporates. So there's halophiles, and then there are the non-halophiles. Non-halophiles can be killed by osmotic stress because of plasmolysis. And so that's why we use salt to pickle foods and sugar to make jams and jellies, because this creates a highly osmotically stressful environment for most of the non-halophiles that may be contaminating food, and it kills them off. It inhibits their bacterial growth. Okay, so that's osmotic pressure and the requirements that organisms have around osmotic pressure. The last is radiation. So gamma rays, X-rays, and even ultraviolet light can damage bacteria because they can mutate the bacterial DNA. Most mutations, when they arise, are damaging to the cell. You know, you don't have a a finely oiled machine and then throw a big old monkey wrench into it and fix it, make it better, generally. And that's what electromagnetic radiation does. It just goes flying through the DNA, ripping its way, breaking up the DNA in bad ways. And so that can even kill bacteria. But bacteria also defend against this kind of damage. They may have pigments that absorb the dangerous wavelengths of energy, that can absorb, say, ultraviolet light, or even X-rays and gamma rays. Or they may have these internal enzyme systems that are very, very efficient at repairing damaged DNA. So, for instance, this guy, Deinococcus radiodurans, I like that name, radiodurans. That's saying that it can withstand. It's durable even under radiation or radioactivity. It can withstand amounts of radiation up to like 10,000 GWs. Oh shoot, I forget what the unit is. But that's the kind of radiation that would kill a human being very rapidly. In part because they carry in their nucleoid regions between 4 and 10 copies of their chromosome. And they have super efficient DNA repair processes that will fix the DNA. And in the meantime, the non-damaged portions of the DNA will continue to make the proteins required for life. And then we use these kinds of radiation to help irradiate foods and kill harmful bacteria that can be on them. So we can use X-rays and gamma rays to help preserve fresh foods like lettuces or apples, or even UV light can be used because we irradiate the food, kill the harmful bacteria that might spoil that food, and help to ensure that it doesn't spoil as quickly. So we can harness the power of electromagnetic radiation to help serve our own ends. Like we humans, we are endlessly resourceful that way. Okay, so there you have it, the last of the physical factors that will affect bacterial growth.